Hello, I'm Dr. Gonzalez, and today in this video we're going to talk about the following topics.
In this video we're going to talk about white blood cells, and white blood cells are also known as leukocytes. So these leukocytes can be divided into granulocytes, which in this page I have the 3 examples, and they can also be divided as agranulocytes. Granulocytes mean that they have granules. Meanwhile, agranulocytes means that they don't have granules. So let's get started with the granulocytes. For this, we have to talk about the neutrophils, eosinophils, and basophils. In terms of neutrophils, I always start with this one because this is out of all of the differential white blood cell count, the majority of these white blood cells are going to be neutrophils. So approximately from 50 to 60 or even 70%, it's going to be neutrophils. And as you can see, neutrophils here are going to be essentially phagocytic cells, right? And they are very fast responders to tissue and destruction by bacteria. They are referred as bacterial slayers because they can kill these microbes by processes called respiratory burst, for example. So again, they can destroy these bacteria by secreting some cytotoxic substances. These substances are often these granules that they have. These granules contain either hydrolytic enzymes or antimicrobial proteins, also known as defensins, right? Let me just go ahead and write it down for you. Defensins. And these defensins essentially will poke holes into the bacterial membranes, resulting in loss of cell content. And they also have the ability to exit blood vessels to enter other tissues. The second one we have to talk about is the eosinophils. Eosinophils, as you can see here, they are not going to be in large quantities. So between 2 to 4, even 4%, right, we could say is eosinophils. They are also granulocytes, as you can see, and they are phagocytic cells. These cells have a nucleus that has 2 lobes, as you can see, and these two lobes are connected by a broad band, as you can see right here, that resembles kind of like ear muffs or something like that. These cells, they have red staining granules. This is why I painted it in red, because these red staining granules contain digestive enzymes, right? So these are going to be cytotoxic substances, and they will be releasing these enzymes on large parasitic worms, for example, so they can basically battle these parasitic infections by digesting their surface. And they also have a little role in allergies and asthma, although the role is not fully understood. And then the third one we have to see here is the basophils. As you can see, basophils are the rarest of all of the white blood cells because they account for only from 0.5 to 1% of all the leukocytes. Essentially, these basophils promote inflammation in damaged tissue, and they release histamine and other chemicals. So basically, in the drawing I made here of the basophils, they have a very large purplish black basophilic granules, right? So you're going to see a lot of these granules within the cytoplasm. These granules contain histamine, like I mentioned before. This histamine is an inflammatory chemical that acts as a vasodilator and acts as attractant to other white blood cells to the inflammation site. So they are going to be functionally similar to other cells that we call mast cells, And because the reason is because they also are very similar in a sense of releasing histamine, and also they are dispersed in the body skin producing this chemical. So one thing that you do in order to reduce inflammation affected, which is increased by this histamine, is taking an antihistamine like Benadryl, because this inhibits or reduces the release of histamine by these cells, thus alleviating the allergies. Now let's talk about the agranulocytes. As you see, agranulocytes, they lack visible cytoplasmic granules, thus they don't have these granules. And there's two types, lymphocytes and monocytes. So let's get started with the monocytes first. Because the monocytes are the largest of all leukocytes, they are approximately between 3 all the way up to 8%. Could be, right, of all the white blood cells. So they are abundant pale blue. They have an abundant pale blue cytoplasm, as you can see here, and this is kind of like a U-shaped nucleus, or U or kidney-shaped nuclei, kind of. So they eventually will leave circulation and enter the tissues, and when this happens, when they exit the bloodstream, they become or transform into macrophages. And as you can see the word macrophage here, macro means big or huge, right? Because these cells transform themselves into very large cells. So they are actively phagocytic cells. They're currently or actively crucial against viruses, intracellular bacterial parasites, and chronic infections. They also activate lymphocytes to mount an immune response, and they arrive in larger numbers to sites of infections, and they take longer than neutrophils to arrive, but they are the ones that clean up the cell debris after they have finished their work. Now, in terms of the lymphocytes, as you can see, these lymphocytes, we have 3 types: B cells, T cells, and natural killer cells, that's the NK. But today I'm going to focus on T and B cells. So as you can see, lymphocytes are the second most numerous white blood cell because it accounts for about 25% all the way, right? In this case, these cells are going to be very large. Also, they have a dark purple circular nuclei, as you can observe here in this drawing I made, with a thin rim of blue cytoplasm, right? So this area right here is going to be a little bit blue. And mostly they're going to be found in lymphoid tissue, for example, like the lymph nodes or the spleen, but a few of these cells can still circulate in blood. They are going to be crucial for immunity, and thus we have these types of cells here. So one of them is going to produce antibodies for removal of specific pathogens and foreign proteins, and others can actively destroy pathogens and damage cells, especially cells that are infected by a virus or even cancer cells. Some white blood cell characteristics that you must know is that some of these white blood cells are capable of a process called diapedesis, which is an emigration process in which these cells can leave the bloodstream, and basically they will be rolling along the endothelium, right? And here I have drawn an example of a blood vessel, as you can see, and they will be attaching to these cells of the blood vessels, the endothelial cells, and then they're capable of squeezing themselves between these blood vessel cells until they reach that area of damaged tissue. The second one is chemotaxis. Kind of sounds like a taxi, right? But It's basically that process when microbes release these chemicals, and the white blood cells are going to be attracted to that invaded tissue by that microbe. So in a sense, it's kind of like they're moving from a place to another thanks to these chemicals. So in a sense, it's like a taxi, right? Transporting them to that place. And then the third one is antigens. Antigens are substances that can elicit an immune response. Often is a protein. And these antigens can be very unique or specific to each pathogen. And thus, white blood cells can recognize these specific antigens, also known as epitopes, for acquired immunity. So now that we just mentioned acquired immunity, let's talk about the differences between innate and acquired. So innate means that you are basically born with it, so you have an ability to resist microorganisms and toxins. Specifically, these innate ability to resist damaging organisms, it's given by your skin, gastric acids, tissue neutrophils, and macrophages, like those cells that we mentioned before, complement microbicidal and lytic chemicals, and these are often in blood and blood cells. Meanwhile, acquired means that you have to basically develop it, and it's going to be a little bit more specific than that innate immunity, because innate immunity is more towards like generic type of microorganisms, and then acquire, it's more specific, and you can divide it into humoral and cellular or cell mediated. So humoral involves more the development of antibodies by your B cells. Meanwhile, cell mediated means that you have to activate these T cells in order to create a specific response.
Alright, that is it for this video. Thank you for watching. Don't forget to subscribe, like, and share. And if you have any comments or suggestions, you can leave them on the comments section. Also, don't forget to check out my Etsy store, Queen Mary Anatomy. That is it. I'll see you next time.